Today we will be studying a very important topic of biochemistry, that is urea cycle. So here is the cycle I'll discuss with you. But before that, let's look at some basics quickly. Urea, as you all know, is the end product of protein or amino acid metabolism. And ammonia produced in the metabolism is toxic. You very well know that. Hence, it is converted to urea. And this urea is excreted out of the body. Synthesis of urea takes place in a very important organ of our body, which is liver. And the site of synthesis is partly in mitochondria and partly in the cytosol. Excretion takes place by the excretory organs in our body, the kidneys. Before moving ahead to the actual urea cycle, you should also know the synonyms of urea cycle or the other names by which we call the urea cycle. Krebs-Henseleit cycle, also called as the ornithine cycle. So these are the two names you must know, which can be alternatively used for urea cycle. Now, urea cycle is a 5-step cyclic process where there are 5 distinct enzymes. The first 2 reactions occur in the mitochondria and the rest of the reactions occur in the cytoplasm. Having understood this, let's dive into the details of this very important cycle. Firstly, as you can see, there is condensation of ammonium ions with carbon dioxide to form a compound which is known as carbamoyl phosphate. Now, this reaction is catalyzed by an enzyme which is called CPS1, carbamoyl phosphate synthase 1 or CPS1. So, this is the enzyme I'm talking about used in the first step. Now, does it matter if I write this as CPS1 or CPS2? Yes, it will make a very big difference. CPS2 is an enzyme used in pyrimidine synthesis, and we are talking about the urea cycle. And CPS2 is a cytosomal enzyme, whereas you can see very clearly here that CPS1 is a mitochondrial enzyme, because this, what I have shown here, is the mitochondria, which I have, whose membrane I have colored in blue. And apart from this, what all you can see is the cytosol. Whereas CPS2 is a cytosomal enzyme. There are many more differences, and we'll not discuss too much of that. Let's come back to the topic. A very important thing to note here is that there are 2 ATPs used in this step, and this is the rate limiting step of whole of the urea cycle. The enzyme CPS1 needs a cofactor which is NAG, that is N-acetyl glutamate. N-acetyl glutamate is a very important name that you should remember. It is a cofactor of the enzyme carbamoyl phosphate synthase 1. And always remember that this step is the rate limiting step. And this enzyme will only be active when there will be the presence of NAG. Okay, the next step also occurs in the mitochondria, and this results in the formation of a compound known as L-citrulline. Let's see how. Citrulline is synthesized from carbamoyl phosphate and ornithine by the enzyme ornithine transcarbamylase. Now, why trans? Trans here is the term used for transfer. Now, what is being transferred? As you all can note, the phosphate of carbamoyl phosphate is here. Now, where is carbamoyl? Basically, this is the group, carbamoyl group of this carbamoyl phosphate that is transferred to ornithine to form citrulline. Can you note one more thing? These red dots, these ones, yeah, these red dots are the transporters. This one here is responsible for transportation of ornithine across the mitochondria from the cytosol. So this is the ornithine transporter, and here, as you can see, is the citrulline transporter, which transports citrulline to the cytosol from the mitochondria. These were the 2 steps that takes place in the mitochondria. Now, what happens in the cytosol? We've already done the two steps, and I told you that urea cycle consists of 5 steps. So next 3 steps will occur in the cytosol. In the third step, citrulline condenses with aspartate, and this results in formation of a compound that is argininosuccinate. So citrulline and aspartate condense to form argininosuccinate. This is catalyzed by an enzyme, argininosuccinate synthase. Since there is synthesis of argininosuccinate, hence it is called argininosuccinate synthase. As easy as that. Now, one more thing to note here is that this step requires ATP. So we noted that first step required 2 ATPs, and this is the third step. This requires 1 ATP. And another thing you have to note here is that ATP forms AMP and not ADP. Now, how will this affect our discussion? I'll tell you that later. Coming on to the fourth step. Argininosuccinate was formed in the third step. What happens in the fourth step? Argininosuccinate is cleaved. So this step is the step of cleavage of argininosuccinate by the enzyme argininosuccinate lyase, or you may simply call it as argininosuccinase. You may simply call it as argininosuccinase. One and the same thing. So this is the other name of this enzyme. Now, what is argininosuccinate cleaved into? It forms L-arginine and fumarate. So basically it forms arginine and fumarate, and this fumarate provides a connecting link with the TCA cycle or the Krebs cycle, as you might remember. Next is the fifth step and the final step, in which the hero of the urea cycle, that is urea, is formed. How is it formed? So here arginine is cleaved by arginase into urea and ornithine, and this ornithine is transported back to this mitochondria from the cytosol by the ornithine transporter. So ornithine is regenerated in this cycle. That is why you might remember, why did I say that urea cycle is also known as ornithine cycle? Okay, so urea is formed. This is the essence of whole of the urea cycle. Now, let's discuss about the energetics of urea cycle. So, we have to come back to the cycle again. In the first step, as I told you, 2 ATPs are used. And in the third step, 1 ATP is used. So, in total, I can say that for one cycle, we need 3 ATPs. But remember, I said that here the ATP is forming AMP. Now that means 4 high energy phosphates are used. 4 high energy phosphates are used. And 3 ATPs are used. So I hope you understood this, because ATP is not forming ADP and ATP is forming AMP. Hence, that is the reason why I said 4 high energy phosphates are used. In the first step, you can see 2 ATPs are used and 2 ADPs formed. Here 1 ATP is used, but 1 AMP is formed and not ADP is formed, right? So that is diphosphate, and here it is monophosphate. That is why 4 high energy phosphates are used. I hope you understood this step. Now, winding up everything, this arginase enzyme, as you can see here, a fun fact just to add on here, this is activated by ions like cobalt and manganese. Now, I'll give you the answer to a very curious question which would be jumping in your mind. See, arginine synthesis, this is the arginine. So, arginine synthesis may occur to varying degrees in many tissues, but only liver can produce urea. Why so? Because arginase is mostly found in the liver. So this enzyme is mostly found in the liver, while rest of the 4 enzymes are also present in other tissues. Got that? Okay. Now giving you a summary, you all know that urea can be written as NH2CO NH2. This is what you have studied in chemistry while you all were preparing for the NEET UG. So here it has 2 amino groups and 1 carbon atom. So from where is it getting these 2 amino groups? For that, we need to come back to the cycle again. So 1 amino group is derived from here, that is the ammonium ion that you can see, or the ammonia. So 1 amino group is derived from here, and the other is derived from this aspartate. And what about the carbon atom? I'll give you 5 seconds to think over it. From where does this carbon atom come in the urea? Yes, you got that right. From the very first step where CO2 enters. So CO2 is the one that provides the carbon atom to urea. Adding to few fun facts here, can you see this ornithine and citrulline here? Do you know that they are basic amino acids? And another important thing is that these are non-protein amino acids because they are never found in the protein structure. Another fun fact is that when we consume protein-rich meals, then that results in increased synthesis of NAG. That is the cofactor required for CPS1. And since NAG synthesis increases, the urea synthesis increases because, as I told you, the first step is the rate limiting step. I think that was all for the video. Don't forget to subscribe my channel. For the urea cycle disorders, you have to wait for my next video, which I'll upload based on viewers' experience and the number of subscribers. Till then, subscribe to my channel and do hit the like button if you found the video worth it.